Yeah, I just turned over the page to start the next set of things. We're starting off with pedigree diagrams. And this is also really important to know for your exam because they can ask you a lot of questions about it. I don't know if they always ask one, but I haven't done one where they haven't asked one, so I think it's a good idea. They'll never ask you to draw one. They just normally ask you, so it's more of an interpretation question. So you need to know, they normally give you a key. So you don't need to know all the stuff off by heart, but there's still some stuff to look at. So they use the pedigree diagram to study the inheritance of characteristics in family over multiple generations. So what they like to do in the exams, they like to give you the genotype of like a couple random people, and then they get you to guess the possible genotypes of the people around them based on those people. Like if they give you the mother's genotype, and then they might ask you to figure out the child's genotype if they're both infected with the same disease or something like that, same disorder. So then you might have to figure out what that genotype is. So that's normally you have to like work backwards. So I'm just drawing it really roughly. They normally print it out nicely for you, so you don't have to worry about drawing it or anything. What's important is that you need to know, but they'll tell you this, that the squares represent men and the circles represent women. So but they'll normally tell you that, for example, the circle is a woman with color blindness and the square is a man with color blindness. So they normally tell you. What's important that they won't tell you is that the horizontal line that connects two shapes means that these people, they use the word mated. That really annoys me, but it means that they had children together. And then if they have, if they are connected by a horizontal line, that means they're siblings. You'll see now. The line that I highlighted there means that they had children together, but the line that like connects them on the top of their heads of the shape mean that they're siblings. And so you normally see there's normally a big gap between like there where I highlighted to show that they had kids together. So you can see the difference between if they were siblings. And then you see that the kids of those two people all have the line that connects them, which means they're all siblings because they have the same parents. Yeah, so that means that they're siblings. Yeah, I do recommend going over this. Yeah, these two at the top are the parents. It normally goes from oldest to youngest. So it'll be parents and their children, and then those people's children. Sometimes they don't do so many, so that it would be first generation, second generation, third generation. They normally only do 3 generations. They don't normally do more than that because then it takes up space. Moving on to mutations. So this is caused by permanent damage to DNA of a cell. They actually get 3 different types of mutations. So they can either be harmless. So this is when non-coding DNA has been damaged. Yeah. So, but non-DNA makes up like 90, some weird number, like 98% of your DNA. Doctors still actually don't know why we have so much non-coding DNA, like what, yeah. But if there's a mutation there, then it's not going to have an effect on anything else because the DNA doesn't code for anything. And then you can get harmful DNA, which is where there's a mutation for a specific protein. So this may cause genetic disorder. So if one protein is wrong, which causes another protein to be wrong. It kind of creates a ripple effect, which ends up causing a genetic disorder. And then some mutations can be useful, which may increase the survival chances of an organism. So it doesn't just mean humans here, it means any organism. And then mutations can occur on genes or chromosomes. So we're going to look at the difference between a gene mutation and a chromosome mutation. So the gene mutation occurs during DNA replication. So it's normally when it replicates for the wrong base. And then this changes the sequence of the bases in the DNA. So examples of this would be hemophilia, which is when your blood doesn't clot. I stand under correction. When your blood doesn't clot. Yeah. Color blindness, difficulty seeing certain colors. I think the most common one is between green and red. And then sickle cell anemia. They like asking about sickle cell anemia. I got that on our mock paper. And then chromosome is non-disjunction, which causes Down syndrome. So non-disjunction is when chromosomes fail to separate, and that causes things like non-disjunction. It doesn't only cause Down syndrome. There's more disorders from it, but that's chromosome. So that happens during meiosis. Now I'm going to talk about biotechnology. The rest of what I'm going to do is not specifically important. So you don't have to watch the rest of this. It's just that this is stuff that we were never told to explicitly go over, but like you'll see at the end of this, I do cloning. We were asked on the sequence of cloning in our mock paper. Like nobody had ever told us to actually study cloning because it's like at the end of the section and they were like, yeah, you can read over it or whatever. And when you tell kids to read over it, they don't. So that so I'm just going to go over these like really simplified and quickly. A lot of it's actually just general knowledge. Um, but nonetheless, it's quite interesting. And they might ask you multiple choice, short questions on it. So it's good to know. So biotechnology is the use of organisms or biological processes to improve the quality of human life. Example, DNA profiling. All the things that I go through now is falls under biotechnology. So I'm going to go over genetic engineering, stem cell technology, and cloning. That's all under biotechnology. I mean, if you look at the word bio, it's like life, and technology is technology. So it's like you're kind of like fiddling around with nature itself.